Dear students, Assalamu alaikum. How are you? Happy? Okay. So, going back again to our studies, if you recall, in our last discussion, so we were discussing about the Golgi apparatus and endoplasmic reticulum, how they are related with each other. Continuing the same discussion of membrane bound organelles. So today we will be continuing with the lysosome, the third membrane bound organelle. Looking at the slide of the lysosome. So what the lysosomes are, these are the vesicles containing hydrolytic enzymes and acid phosphatase. Digestion of food is an important function of these lysosomes, which is engulfed by the cell. Digestion of foreign particle is another important function, and they are concerned with the autophagy, autolysis, self-eating. If the cell has completed its life, so that is also being broken down by these lysosomes. And the last, exocytosis, the product of lysosome, they are exported outside the cell by this process of exocytosis for the use of other cells. These lysosomes are having a very, very important function because they are having hydrolytic enzyme and acid phosphatase. So what is a hydrolytic enzyme? So you must be knowing that these are the enzymes which are concerned with hydrolysis. What is hydrolysis? Breakdown of the larger compound into smaller compounds. So we are eating food in the form of carbohydrate, proteins and lipids. These are macromolecules, big molecules. So these are going to break in the small molecule for the purpose of absorption into our blood. So lysosomes, they are having hydrolytic enzyme to convert carbohydrate into monosaccharide, proteins into amino acid, lipids into fatty acid and glycerol. So they are concerned with the digestion of food. It is not only the extracellular digestion, so you may be knowing about some unicellular organism like amoeba, like paramecium. So these cells are going to take the food by endocytosis. These lysosomes get fused with that food vacuole and discharging their hydrolytic enzyme, causing the digestion of that food material inside. So this is the intracellular digestion. So extracellular digestion, the enzymes present in the lysosomes are secreted in the form of gastric juice, intestinal juice, bile, pancreatic juice, all of these. So these are the products of lysosomes. While intracellular digestion in case of unicellular organism and the certain cells of multicellular organisms also. Another important function, we are exposed to lot of pathogen. We are inhaling virus, we are taking a lot of bacteria through the air and in our food. If you are going to go to a vendor outside and just enjoying golgappes, so what is there? A lot of germs are going to go because that person is not aware of hygienic preparation of the food. But you are not having diseases. Why? Because these are the lysosomes given by the God in our white blood cells, the phagocyte, neutrophils in our blood. So that virus, that bacteria, that germ is going to enter in our body and the WBCs are going to take care, they are going to have the phagocytosis of that inside, and these are the lysosomes. They will go there and they will attack that virus, bacteria, releasing their hydrolytic enzyme and acid phosphatase, breaking the body of virus, bacteria into small component, and that is being dissolved. So this is the immune system given by the God that is working on the basis of lysosomes. Another important function is that every cell, like every one of us, is going to die away. We are having a specific period of life to play our role in this universe. Similarly, every cell is having a specific life. There is an inbuilt program of cell death. Every cell is having an inbuilt program that after so many days, the cell is going to die away. Like, what is the life of a RBC? All of you must be familiar from the lower level, from your metric, O levels, FSC levels. So you must be knowing that the life of RBC is 120 days. What is the life of WBC? Because they are the fighters. They have to fight with others. They are being killed. So life of WBC is 3 to 4 days. Life of platelets, 7 to 8 days. Every cell is having its life. And once that point of the time of death is going to reach, so what is there? These are the lysosomes inside the cell. They rupture. And once they rupture, these hydrolytic enzymes, they are going to scatter here and there to dissolve whole of the cell. This is called as autophagy, self-eating, autolysis, self-breakdown. So these are also called as suicidal bag. If the cell want to commit suicide, it means that all the toxic compounds, they are present inside the cell, made by God at the time of birth of every individual. So they are concerned with the process of aging. So this was the lysosome. Looking at one of the animation, showing you some of the processes that how the lysosome is going to act. Looking at the functions of lysosomes. So these are the lysosomes having these hydrolytic enzyme insides. So these are the hydrolytic enzyme for the breakdown of any variety of organic compounds, that is the carbohydrate, proteins and lipids. So this is the, let's say, the Golgi apparatus. This is the endoplasmic reticulum, where the various products they are being produced. These endoplasmic reticulum giving their product to the Golgi apparatus in the form of transport vesicle, finishing and packing of the product, and these are the lysosomes. Now this is a bacteria which is going to enter inside. These are the bacteria and they are attacked by the lysosomes. And what is there? Oh, splitting of the bacteria. They are going to be disintegrated, they are going to disappear. Let's say this is the mitochondria which have completed the life. Autophagy, self-eating, the part of cell is digested. Similarly, whole of the cell may be digested at the time of death of the cell. So these are the few functions of the lysosome you have seen in this animated tutorial. Going with the two more membrane bound organelles, that is the peroxisome and glyoxysomes. They are again nothing special. These are the membrane bound small vesicles, but instead of hydrolytic enzymes, they are having different variety of enzyme. So what the enzymes are present in these and what they are going to do for us. So looking at the slides, peroxisomes. So peroxisome, they are bounded by a single membrane, and they are having they are generating and degrading hydrogen peroxide in performing various metabolic functions. So they are not formed from endomembranous system, but by incorporation of proteins and lipids from the cytosol. Similarly, going with the function of these peroxisome, vesicles containing oxidative enzyme like catalase. So this is the enzyme catalase which is present. This enzyme is catalase, and any you can say the waste material, that is the hydrogen peroxide, it is detoxified into water and the oxygen. And most of these peroxisomes, they are present in liver and kidney cell to deal with the toxic molecules. So these two organs, liver and kidney, they are assigned the role of detoxification. Similarly, exactly the same glyoxysome, they are bounded by a single membrane, but the enzymes are different. These enzymes, they are glyoxylate enzyme. And what glyoxylate enzymes are concerned? They are concerned with the conversion of lipid molecules into carbohydrate. And this function is required especially in the fat storing tissues of germinating seeds of plants. These two organelles, that is peroxisome and glyoxysome, so they are again very vital. Whenever we are going to have any toxic compound in our body, so mainly the liver cells, to some extent the muscle cells also. So they are going to detoxify that, and during this process of chemical reaction, usually a waste product, hydrogen peroxide, is produced. Hydrogen peroxide is a very toxic product if it is going to accumulate. There is a need, either it must be given out or it must be converted into less toxic form or non-toxic form. So peroxisomes, they are having the oxidative enzyme, the catalase, and this catalase is going to break down the hydrogen peroxide into water and oxygen. Both of these are harmless. So this is a detoxifying function. With the reference of the second organelle, that is a glyoxysome, having these glyoxylate enzyme, so seeds are kept to have the minimum weight. Why? So you must be knowing that dispersal of seed is a very important topic. So God has produced the seeds that they may be dispersed in a wide variety of habitat. To keep the weight minimum, if the starch or storage of carbohydrate is there, so that is going to require double the amount of storage. In comparison to that, oils and fats, they are going to store a large amount of energy within a very small volume. So these are usually fats which are stored in the seeds. Once the seed is going to germinate, so what is there? It is going to require energy because there is no photosynthesis at that time. So these fats and oils which are present in the seed, they need to be converted into carbohydrate because we can generate energy from the carbohydrate. So these glyoxylate enzymes, they are richly present there, so they are going to convert the fat into carbohydrate. This is called as glyoxylation. So glyoxysomes, they are more commonly found in case of plant cell. Going with our next membrane bound organelles, that is the mitochondria and chloroplast. Both of these are bounded by the double membrane. They are different from others. Up till now, whatever we have discussed, they were bounded by the single membrane. But these two, mitochondria and chloroplast, they are bounded by the double membrane, and every structure is made by God according to the function. So there is a requirement that there must be double membrane. So looking at the structure and function of these mitochondria and chloroplast. So this is a view of mitochondria. These are present in both the animal and plant cells, and they are given the job of releasing the energy. So the energy is released by the breakdown of the food, usually the glucose, resulting into carbon dioxide and water. And these mitochondria, they are also called as powerhouse of the cell because they are going to supply energy for any purpose. Energy is required, so these are the mitochondria which are going to provide energy. And those cells which are active, they are requiring more energy, like muscle cells. So they may be having more than 20,000 mitochondria per cell in every cell. But those cells which are requiring less energy, they may be less in number. So looking at the structure of the mitochondria, so there is different compartments inside. As I was just give the information that they are double membrane bound. So this is the outer membrane which is very smooth. This is the inner membrane. And the inner membrane is folded. So this is one fold, another fold, another fold, another fold. Between these two membranes, there is inner membrane spaces there, and these folds of inner membrane, they are called as cristae of mitochondria. Cristae of mitochondria. Inside these cristae, so there is a liquid material, a matrix of mitochondria which is going to have a lot of enzyme. This is a electron micrograph of the mitochondria, the outer membrane, inner membrane, and these are the folds of inner membrane called as cristae of mitochondria. So they are again another view of mitochondria. Another view of mitochondria, more clearer view, the outer membrane smooth, inner membrane folded. These folds are called as cristae of mitochondria, and this liquid which is there inside, this is called as matrix of mitochondria. the role of mitochondria in provision of energy. So basically glucose is going to break down into two molecules of a 3-carbon compound called as pyruvate, and this reaction is called as glycolysis. This is occurring in the cytoplasm of the cell, not requiring mitochondria. If oxygen is absent, then this pyruvate is not allowed to enter in the mitochondria, and this pyruvate is either converted into lactic acid or into alcohol, and that is lactic acid fermentation or alcoholic fermentation, and this is called as anaerobic respiration. If oxygen is present, then the receptors are present on this outer surface of mitochondria. They will detect that this is the pyruvate and oxygen is present, okay, allowed to enter. The gates are open for the pyruvate, and pyruvate is going to enter in the matrix of mitochondria. And in the matrix of mitochondria, a reaction called as a link reaction. All of you must be familiar with this reaction at your lower levels. So you must have studied the conversion of pyruvate into a molecule of acetyl CoA with decarboxylation and dehydrogenation. This is called as link reaction. And then this acetyl CoA, which is produced by this link reaction, it is going to combine with the already present 4-carbon compound in the matrix of mitochondria called as oxaloacetate, and this oxaloacetate is being converted into citric acid, alpha ketoglutarate, succinic acid, fumaric acid, malic acid, and again reproducing oxaloacetic acid in the form of a cycle which is called as Krebs cycles. And in the Krebs cycle, only one ATP molecule per Krebs cycle is produced, with very little energy, but most of the energy is given in the form of reduced NAD and reduced FAD. So these NAD and FAD, they are given to the cristae of mitochondria for oxidative phosphorylation, and all of these, they are converted into ATP through this process of oxidative phosphorylation or electron transport chain. If oxygen is absent, only 2 ATP, 2 energy molecules are produced in anaerobic respiration, and if oxygen is present, then this mitochondria is going to produce 38 ATP molecules from one glucose. So this is the role of mitochondria, 38 ATP molecule. In comparison, if there is no oxygen, no role of mitochondria, only 2 ATP molecules. Looking at one of the animation to see the role of mitochondria, how they are going to provide us energy. So this is one of the mitochondria having the outer membrane, inner membrane. These are the cristae of mitochondria. This is the inner membrane, and these are the NAD and FAD as I was mentioning. So they are being brought on these cristae of mitochondria which are located on the inner membrane, and these NAD and FAD, they are going to pass through the chain of these electron carriers. So these are the electron carriers. They are going to pass from a higher energy state to a lower energy state to coenzyme Q. Splitting of the electrons and protons are there, and from coenzyme Q, so they are given to the further carriers, so that is BC2 complex, and further modification is there, and then they are given to the cytochrome C to another carrier, and from the cytochrome C, so they are it is the FAD which is going to pass through both of these stages, and both of these, they are also given through the coenzyme Q and through this, you can say, cytochrome C, and further oxidation is going to occur here. The protons are going to leave outside, and then it is a proton gradient in the inner membranal space between the outer and inner membrane. So these are the protons, and these protons now they are going to combine to make a combine with the oxygen and these electrons to produce a water molecules. So whole of this is going to operate on the cristae of mitochondria, and this is going to occur in a rapid phase. These protons are going to pass, and they are going to convert this ADP into ATP. This is the energy which is synthesized by this process, ADP converted into ATP. So this is the proton gradient which is going to generate ATP. Dear student, this was the story of powerhouse of the cell, how the energy is generated. Now going with a reaction which is almost opposite of this reaction. What was that reaction in mitochondria? It was one of the catabolic reaction, breakdown reaction. Glucose is going to break in carbon dioxide and water, releasing energy in the form of ATP. So what will be the opposite of that? Anabolic reaction. So plants are given a specific role that you should be synthesizing food, and this food is synthesized by one of the organelles in the plant cell, and that is called as chloroplast. It is not only the chloroplast, but there are many other membrane bound organelles which are present in the plant cell, which are having pigments of various variety. So we are collectively using a terminology of plastid. Plastid is basically defined as membrane bound pigment containing organelles. Chloroplast is one of the famous plastid, the most of the students are going to know, but we will be looking with the other variety of plastids also. So going with these plastid, they are found only in plant cells, and three types of plastids are there. Chloroplast, they are green in color and they are used in the process of photosynthesis. The second variety of chloroplast are called as leucoplast, and they are concerned with the storage of food and starch in case of plant cells, especially in the parenchymal cells. Then these are the chromoplast, and they are going to store the pigments or colors. Chromoplast. So you are going to see the beautiful colors of petals of flowers and fruits, and most of the other parts of plant, they are green. So these different colors, they are given by the chromoplast. The pigments which are stored, they are called as anthocyanins. So you must be enjoying the beauty given by God. So one is the greenery, and then these are the various colors in the kingdom plantae. So how the same single chloroplast is going to give us the various variety of color. So all of you must be familiar with the printer. Many of you will be having at home the printer. So what you are using in the printer? A ink cartridge for black and white, and one is the color cartridge. And for normal printing you are using black and white, common printing. But sometimes you are having the color print, and this is the single ink cartridge which is going to give you all the color, green color, yellow color, orange color, everything. Similarly, the God has given the greenery, the common color in the kingdom plantae, and this green color is due to chloroplast. This is the chlorophyll which is of green color present in the plant. And just like our color cartridge, chromoplast are given by God. They are present in the petals of the flowers and in the fruits. So they are going to give all other color with the exception of green colors. So these are the plastid, but we will be mainly focusing our discussion on the chloroplast because they are concerned with a very important process of photosynthesis. So looking at some of these slides, this is one of the plastid, the chloroplast. As I was mentioning, that both the mitochondria and chloroplast, they are double membrane bound. So this is the outer membrane of chloroplast, outer membrane. And then this is the inner membrane, and there is a space called as inner membrane space. This is the space, inner membrane space. So in the mitochondria, the inner membrane was folded. Here in the chloroplast, the inner membrane is also folded in the form of sacs. So this is one sac, and this single sac is called as thylakoid. And if you are going to look, so these are just like coins. This is one thylakoid, below that it is another thylakoid, below that another thylakoid. So this set of sacs, set of thylakoid, it is given the terminology of grana of chloroplast. Grana of chloroplast. So these granas, they are also connected with each other. These are called as intergranal lamella. Intergranal lamella. So looking at the role of the chloroplasts, so they are green organelles, and they are assigned the main function of synthesis of food. They are found only in the plant cells, and their main role is they are capturing the sunlight and the energy which is there in the light, it is going to fall on the chloroplast, and that energy is the light energy which is converted into the chemical form of energy. In the presence of this chlorophyll in the chloroplast and light, carbon dioxide is going to combine with water to give us sugar and oxygen. Oxygen is the waste product of this reaction, but very useful for aerobic organisms, and sugar is the main product. These are the carbohydrates which are made by this process of the chloroplast. So these chloroplasts are given very important role, and these important role, that is to manufacture food by the process of photosynthesis, and that is occurring in the two steps: light dependent reaction occurring on the thylakoid and the grana of chloroplast, and light independent reaction, the Calvin cycle, which is going to operate in the stroma of chloroplast, in the liquid part of the chloroplast. Looking at an animation to have the role of these chloroplasts in the process of synthesis of food. Looking at this animation, so you can see the water and carbon dioxide, they are going to enter, and this is the light which is going to fall on the chloroplast, and the two products, they are being produced. So this is the entry of water and carbon dioxide, light falling, and the glucose and oxygen, they are being produced. So in this program we discussed about some of the membrane bound organelles, that is the lysosome, peroxisome, glyoxysome, and the last two, mitochondria concerned with catabolic reaction, and the chloroplast for anabolic reactions. So inshallah, in our next program we'll be continuing with the discussion of the rest of the organelles in the cell and how they are going to perform the special function for the continuity of life. Till that time, Allah Hafiz.